It's a pleasure to be here. I think Dr. Levy and I have complementary points of view. I'm going to talk about a pathologist's view of personalized precision medicine, and then she'll give the other view. I'll summarize my disclosures is that I do a lot, but I don't get any money. But I am available. So let's start out with kind of the where we were and where we are now. So when I was a resident, I used Anderson's Pathology, and the WHO classification of lung cancer had acinar papillary bronchoalveolar and solid with mucus formation. In the 21st century, we have a new WHO with more words, and now let's have a kind of a look at what it is. So acinar is still acinar. Papillary now is papillary and small papillary, micropapillary. Bronchoalveolar, the words didn't ring, so now we have lipidic and mucinous, former mucinous bronchoalveolar, kind of like Prince. And then solid with mucus formation is solid with mucin production. So the conceptual framework for adenocarcinoma of the lung in the many years since I was a resident and now really hasn't changed. And that's really made evident by this bid on eBay for the book that I used. It's worth $3.80. And to a certain extent, that's a bigger statement than just what the book is worth. In modern pathology, it's great to be a morphologist. It's great to be perfect and get them right 100% of the time, but it's worth about $3.80. Now, pathologists have many critics, and Steve Friend cloned the retinoblastoma gene in Robert Weinberg's lab, and I won't read the whole thing, but he calls us the shamans of medicine. He says that, you know, we pickle tissue, we put it in wax, we stain it with extracts from roots and berries, we give it complicated names, and we ignore modern technology. And the truth is, we're living in a world of genomically informed medicine. Now, what you're all here to think about and talk about is next-generation sequencing. And clearly, next-generation sequencing is a powerful tool. It's kind of like maybe the Colt pistol that God made all men equal, but Samuel Colt really made all men equal. But you come to a point where one technology isn't necessarily the complete solution. And pathology we come back to as the fundamental foundation for diagnostics. And so now kind of my major message, one that will ring through, is we have to find the right technology. We have to put the right technologies together and use them in concert. And we have to present that information in one package. So this is one of my favorite slides. My wife always tells me not to show it because it's stupid. But you see three men with a task, right? And the whole idea is how to choose the right technology to fit that task. Each of them have powerful tools. So we have genomics, we have pathology, and how do we bridge the gap? And the gap is really informatics and bioinformatics. And if you think about how we're deluged with information and we're deluged with a variety of different kind of technologies, think about a clinician who gets 10 different reports that need to be condensed into one diagnosis and one treatment plan. So I alluded to this before. If I have a perfect pathologist, somebody who is, you know, Rosai or better, they can get the right answer 100% of the time, but it will never lead to the patient getting a targeted therapy. So we need to know the disease genotype and the signaling pathway, and the classic way to do that is with a companion diagnostic. And up to now, companion diagnostics have been one by one, right? So BRAF had a BRAF test, EGFR, KRAS. They were all individual tests. Now it's clear that the paradigm has changed. And with the MATCH trial, we now know that a genomic classification of disease is really critical. So let's talk a little bit about adenocarcinoma of the lung. And let's think now about the testing and the different modalities that we use. NCCN guidelines say that for disseminated or advanced stage adenocarcinoma of the lung, we should do EGFR and ALK testing. And this is a couple years old. And then if you summarize their recommendations, they actually recommend EGFR and RAS testing, KRAS testing. We look for sensitizing mutations for tyrosine kinase therapy. We look for resistance, and KRAS has been associated with resistance, both primary resistance and in some instances secondary resistances. And we look at the ALK gene fusion. So we do PCR or next generation sequencing for EGF receptor and KRAS, and we do FISH for ALK. Now, ASCO in late August came out with new recommendations, and they really are a bit of a game changer. So the first of all, they recommended that all lung cancer samples, adenocarcinomas, be tested for ROS. And that was based on what appears to be favorable responses of ROS, cancers with ROS gene activation to crizotinib. They recommend EGFR mutational analysis, but they put a couple qualifiers in. They want the analysis to include all sensitizing mutations that have a frequency of greater than 1%. So now we're in the realm of next generation sequencing, right? And then they want all of the mutational analysis, even at the time of diagnosis, to include resistance mutations. Now, the other interesting thing is they've taken a step back. NCCN suggested that the testing be done on advanced stage. Now they say, you know, it's okay to test early stage. So you don't have to wait for stage 4 patients. You can test patients at earlier stage. The other interesting thing is that they recommend a full genomic analysis of samples in small biopsies that appear to be squamous, but you can't exclude the presence of an adenocarcinoma. And they also recommend testing in all cases that may have a mixture, so adenosquamouses. And finally, one that I didn't put up there is they give a turnaround time. They say that testing should be done within 2 weeks, 10 working days. So what do we do at Jefferson? So I want you to look at the bottom part of the slide. For adenocarcinoma of the lung, we do next generation sequencing on a panel. We report EGFR. we have the guidelines in that we do all mutations with a frequency of greater than or equal to 1%, and we do resistance mutations, and we do KRAS. And I can tell you on many Friday afternoons when I sign out cases, and it's a small biopsy, and I'm looking at EGF receptor, and it's wild type, my blood pressure goes up because I wonder, you know, do we have enough tumor? Is the sample big enough? And if I see a KRAS mutation, my blood pressure goes back down. So now we do those two. If those are negative, we do FISH for ALK, ROS, and RET. And if those are negative, we do BRAF, extended BRAF, and as you know, 50% of the mutations are in codon 11 or exon 11, 50% are in exon 15, and we look for the insertion in HER2. Our philosophy has been all of those genes are actionable, so it's important to do those for our patients, and we've been reporting these separately up to now. So to a certain extent, this is the kind of picture that brought us to the dance with GenomOncology. How can we package the information from different technologies for one diagnosis for the patient that gives the oncologist one set of information and puts it in one place. Okay, so let's have a first clinical example. This is a patient, my eyes aren't as good as, can you guys see that any better? This is a patient with an ALK rearrangement, and so let me show you here. It has positive for an ALK rearrangement, and it has a point mutation that's of undetermined significance and almost certainly of no significance in P10. So we have an integrated results summary where we give the high points. We have a box for interpretive statement. Then we have essentially the pharmacogenomics of what genes or what mutations are associated with sensitivity to a pharmacologic agent, what's associated with resistance to a pharmacologic agent. And then we have a list of clinical trials. After that, we give each report separately, so we have the full body of the report and we fulfill CAP requirements. So we have fluorescence in situ hybridization, and for each report, we show relevant images. As we're doing it now, we show all positives and all negatives. So this would have ALK as positive. This is ROS and RET. We don't show an image, they're negative. And I think here they weren't tested. So let's show clinical example number 2. This is a non-small cell lung cancer with EGFR mutations that's sensitizing and resistant. So here we have, again, our integrated results summary where we show that there's an EGFR. I think this is exon 19, the common deletion. And then there's the 790 mutation. Then we have a P53 mutation, and it lists the genes that are negative. Since our testing algorithm says if it's EGF receptor positive, we won't burn more of the patient's or the hospital's money, we don't have the rest of a composite report because we haven't done those tests, because essentially they're mutually exclusive. So again, pharmacogenetics here showing that the 790 mutation is associated with resistance. And I believe these are the cumulative report. And this, again, lists the mutations and the resistance. We also have a list of clinical trials. So there's guidance for the clinician on what clinical trials are available. After that, and I haven't included all of this, we repeat the mutational analysis as it would be in a report. We think it's important to show actually examples of the mutations. There was an article in Nature Medicine about good genomic practice. And so here we show that there is a deletion. There's no strand bias. You can see it in forward and reverse. Here we show the point mutation at 790 and a description of that. And here we show the P53 mutations. And as you know, P53 is a very common mutation in lung cancer. Now, one of the challenges that we have, and again, how do we put together a report and how do we allow space and capacity for different kinds of diagnoses? So just for interest, here's our testing algorithm. One of our challenges, and I've heard this in multiple settings yesterday, is small transbronchial biopsies or lavage. So we start out with histopathology and cytopathology. We go to next generation sequencing for EGFR and KRAS and the other genes in the panel. If we don't get readable material or sequenceable material, we can reflex to immunohistochemistry. And so we've had early access to monoclonal antibodies to the exon 19 deletion, to the exon 21 point mutation L858R, and to ALK. I'm going to show you some examples of these. And together, those two EGFR mutations account for about 80 to 85% of sensitizing mutations. So even in the presence of a small biopsy, we have a good chance of detecting a sensitizing mutation. So this is the anti-EGFR exon 19 deletion. This was prepared to the 15 base pair deletion, which is the most common. This is not perfect if it's not a 15 base pair deletion and goes up to 18 or 21. Sometimes we have false negatives, but for the 15 base pair deletion, the activity is quite nice. And so here's the case. It was a very small biopsy with a nest of malignant cells. They stained for TTF1, and you can see quite nicely that they stain with the antibody for the exon 19 deletion. This is another case. You can see that we had fibrous tissue, nests of cancer cells here. You know, this is hard to sequence out of paraffin. And here you can see quite nicely that there's staining for the L858R. And our correlation between sequencing and reactivity with this antibody is almost 100%. And then finally, we got early access to the Ventana antibody. We actually used the clone from Spring since the Ventana antibody was in FDA trials. We validated that again against our FISH. This is an interesting patient from Europe who had sequencing by a prominent company that does huge next-generation sequencing. They showed an ALK fusion, but she wasn't eligible for a clinical trial because the clinical trial for the second-generation drug required FISH or immunohistochemistry. It was positive for FISH, and you can see, even though it looks pretty poorly differentiated and solid, that it's quite strong for expression of ALK protein. So we now have developed a format where it's modular. We can accommodate all of these technologies with a consolidated report, starting with the summary, an interpretation, and then the individual reports in a queue. Another disease that requires multi-modality testing is AML. And now let's look at the phases. So at diagnosis of AML, we do a bone marrow, we do cytogenetics, we do flow cytometry to establish the diagnosis. The patients get induction therapy, and induction therapy is the same for all patients except for one type, which is acute promyelocytic leukemia. So we do that by FISH, and we do that with a same-day turnaround. So it's done very rapidly, so we can start induction therapy. The patient achieves a hematologic remission. There's a gap between the initial diagnosis and the remission of a month to 2 months before consolidation therapy starts. And then tests that we do have an impact on what consolidation therapy is. If it's a low risk, the patient might get chemotherapy. If it's a high risk, the patient can get an allogeneic stem cell transplant. So again, we have a panel that we do by next generation sequencing that includes nucleophosmin. If it's mutated or CBP alpha is biallelically mutated, it's a more favorable diagnosis or more favorable prognosis, so the patient gets chemotherapy. If it has a FLT3 internal duplication or mutation, it's probably a poor prognosis, and the patient would go to allogeneic bone marrow transplant. So here, in contrast to lung, where we have predictive tests that lead us directly to a therapy, here we have one predictive test, APL. It's done right away by FISH. We have other tests that are done genetically. And these are predictive tests, but they're actionable, and they give rise to different therapeutic approaches. So this is, again, just kind of a review for good prognosis. We have fish, 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 but we have mutations as well. So we need multiple technologies to develop a mature therapeutic strategy. This is our next generation sequencing protocol. We've developed this panel in-house and validated it. It has the 4 genes that are category 2 evidence by NCCN guidelines. These are the ones we do. These are the ones we charge the patient for. We also do these, and we report these. We're developing a protocol with the clinicians, so we do these tests. There's not immediate medical relevance, but the patient has been consented that we're doing additional genetic tests. And that shows hotspots. So the third clinical example and the final clinical example is a case that was called AML by our hematopathologist. This was a case where, it was, there are a few times where it's good to be old. So it had a little bit of fibrosis and it had blasts, and they said it's AML, and I said, guys, it's CML and blast crisis, and they didn't believe me. Fidel Castro said, condemn me, it doesn't matter, history will absolve me, so I got absolved on this one. And here's our report. So you can see that we have in situ hybridization that's positive for the fusion. We have cytogenetics, chromosome analysis, that's positive for the Philadelphia chromosome. And by our mutational analysis, we show that there's the classical resistance mutation in ABL1, in ABL. There's a space for prognostic information, pharmacogenetics that suggests third-generation drugs that might be active, and drugs that may be associated with resistance. Evidently, there aren't clinical trials for this. In the report for the genetic analysis, it gives a description of the mutation, so we have an educational component for the clinician. And this is the now mature report for chromosome analysis. We include an image of all positives and negatives. We have the fluorescence in situ hybridization showing that it's positive. And then we have an image, and I apologize, this is small and you can't see it, but it showed this is a double fusion and deletion of the region on ABL, right by ABL. It's called ASS. So in summary, precision diagnostics includes diagnostic, predictive, and prognostic data, and the therapeutic decisions require a spectrum of techniques that give us either personalized therapies, as the case that I showed you in the leukemia, or targeted therapies, as the case that I showed you in lung cancer. Comprehensive reporting must accommodate pathologic diagnosis, and my determination is that I always put the pathologic diagnosis in the field using the exact same words that the pathologist used. Then we have cytogenetics, fluorescence in situ hybridization, molecular analyses, next-generation sequencing, and then also potentially immunohistochemistry. Since the process is modular, it fits all of those. And the comprehensive reporting provides a framework to promote appropriate utilization. If you start reporting everything together, you can go through kind of a learning process with the clinicians in what really contributed and what didn't contribute, and how you work through this. So I'm within my time, happily, and I'll leave you with the story of Procrustes. And Procrustes, in Greek mythology, was a son of Neptune with, I think, a nymph, a sea nymph, and Procrustes was kind of a thug who hung around outside of Athens. And Procrustes said he was an innkeeper. And so if he captured you, he would take you back to his inn, and you had to sleep in his bed. Now, the trick was you had to fit his bed perfectly. So if your legs were too long, he cut them off. And if they were too short, he stretched them. And supposedly nobody fit perfectly into Procrustes' bed. And so the modern pathology has to accommodate technologies. We can't, fit into the old diagnostic boxes. And Theseus, as one of his kind of superhero feats, one of his tasks, captured Procrustes and killed Procrustes. So I think genome-oncology can help us be a pathology Theseus, put all the information together and not fit into the old boxes. So thank you, and I'll turn it over to Dr. Levy on time.
Well, I know I can't beat the analogies, but I think Dr. Peiper really set me up very nicely for what I want to talk about is how we're enabling these comprehensive interpretive reports and these multimodality interpretations through curation efforts in My Cancer Genome and a new framework for how we represent that knowledge being enabled by GenomOncology's new knowledge management system, which we're co-developing. So as was very well articulated, as a medical oncologist on the receiving end of your reports, I'm now faced with this massive amount of data coming into the clinic at an unparalleled rate that we've never really seen before. So we really evolved from these single mutations to the hotspot panel testing, now to next generation sequencing assays. And the clinicians are getting a tsunami of information coming into the clinic. And the challenge, the reason why this is challenging is because the data is coming in at multiple levels of evidence. Depending on how big your panel is, you may only have evidence that's at the level of the preclinical significance. There perhaps has some clinical validity in the sense that it separates one population into two or more groups with distinctly different outcomes, but I may not actually be able to use that in my clinical care, which is really where clinical utility comes in, is that it has to meet that criteria whereby I'm actually going to make a diagnostic or therapeutic decision differently. And all of this data is coming in at multiple levels of evidence with different types of studies, including evidence just from case reports that we're now using to try to make clinical decisions because there just isn't enough of a body of knowledge to know what to do in every situation. So we were inspired by this problem back in 2009 when we started our own multiplex testing at Vanderbilt, and this culminated in the creation of My Cancer Genome, the ongoing mission of which is to curate and disseminate knowledge regarding the clinical significance of genomic alterations in cancer. As many of you know, My Cancer Genome is a publicly available website. You can go and search for information now with new information on 21 different cancer pathways, over 700 different cancer genes related to those pathways, and then detailed curated information on 21 different cancers, which are listed here, which have high clinical significance, and over 400 variants and over 500 cancer drugs, complete with information on their targets. This content has grown over time, and in about 2 years ago, we started having discussions with GenomOncology on how we could leverage this information beyond just our website to be able to provide information to clinicians at the point of their interpretive report. But in order to do that, we had to undergo some significant re-architecture of the back end of our resource. And together we've been moving forward to create a new paradigm. And I'd like to share with you some of that idea. So some of the things that you will be aware of is that the clinical knowledge is not just represented in variants. You have point mutations, insertions, and deletions, but some of the knowledge is represented at the level of the exon. And so you need to be able to say something that's related to the clinical literature about an exon, but when you actually go to have a patient who has a particular variant within that exon, to be able to map it back to the knowledge for that information. We cover information on fusions, rearrangements, gene amplifications, and even protein expression. And I should add, the cytogenetics has become one of our newest areas. I need to add that to my list. And it gets more complicated than that because these biomarkers are not in isolation. They're often co-occurring alterations that need to be taken into account. And when we first started out developing My Cancer Genome content, we were really in that era of, a single point mutation predicts sensitivity to a drug, but that just isn't the case anymore. So we've had to evolve our representation of the factual statements that we're making from the literature, and we have several different components of therapy assertions, prognostic assertions, and diagnostic assertions. And so for an example of a therapy assertion, you have the example you gave earlier with the single alteration of the EGFR L858R mutation, which in isolation would likely confer sensitivity to EGFR tyrosine kinase inhibitors such as erlotinib in the metastatic setting. But if you have an EGFR T790M mutation co-occurring with that sensitizing mutation, you very likely have an acquired resistance to that same drug. When you see reports that only talk about these variants in isolation, it can be very confusing to a clinician to try to understand how to interpret what they're seeing when one says sensitive and the other says resistant. And the names are kind of confusing too. No one's going to memorize T790M and L858R. So we need tools that help us put those pieces of information together so that the comprehensive interpretation is that the patient's likely going to be resistant to this therapy. And then, of course, it gets even more complicated when you get all the not assertions, things that shouldn't be present in combination with one another, no exon 2, 3, or 4 mutations in KRAS or NRAS in order for a colon cancer patient to be sensitive to cetuximab, for example. And on top of these complicated and not relationships, the level of evidence for the various sources has evolved over time, and they sometimes don't quite catch up with one another. So the FDA package inserts are quite limited. They have their initial approval for an indication, and usually it's a single companion diagnostic, as you mentioned, that is driving the approval of that drug, but they often don't update those guidelines or those indications in their package inserts, whereas the NCCN and ASCO are continuously updating those at a more rapid rate. So you need to be able to take into account that the knowledge is growing over time and what resources can we use to do that. So those are some examples of these therapy assertions. We now have over 180 of these therapy assertions, which have been created to help drive, with work with GenomOncology, to drive the interpretive report in that therapy interpretation section, which Steve just showed you. In the prognostic space, we're now, as I mentioned, getting well beyond traditional My Cancer Genome into things that are broader in a biomarker space. And using AML as a very good example, if you were to look at an assertion in isolation for a normal karyotype in a patient with AML, you used to say that would have been intermediate prognosis. But now that we have all of these other biomarkers that need to be done in conjunction with karyotype, you really can't say much about karyotype in isolation by itself. And so we've evolved that to now say it's more of an indeterminate prognosis. Whereas if you add karyotype with an NPM1, let's say exon 11 mutation, now you may have a favorable prognosis in the setting of those two co-occurring alterations. But then make it even more complicated, and now you add a FLT3 ITD, and now you've taken that favorable prognosis into an unfavorable state. So understanding that all of these assays are important, all these tests are important, whether they're done individually or they're done in a multiplex fashion using NGS or other technologies. We need to put all of these together into these combined rules that help us understand what interpretation to help the clinician understand, to know what to do with the information when it's brought together. And so we now have, I believe, over 120 of these prognostic assertions added into the knowledge base. So if we take that framework that we've just been describing of how we group alterations together as groups of alterations that can co-occur, we've also started to extend this to our understanding of the clinical trial space. So Steve mentioned the NCI match trial. Who's familiar with the NCI match trial here? Almost everybody. Great. So, the NCI match trial is this very revolutionary basket, umbrella, tent, who do you know what to call it, trial that has basically got 21 different arms, each of which is going to stratify patients to a different molecular profile based on their next generation sequencing results. And in order to appropriately stratify patients, each of these arms is going to have a different set of eligibility criteria. And so you'll have a set of diseases that are either included or excluded, plus a set of biomarkers that are either included or excluded. Then, of course, you have all of the other things that help a patient become eligible for a trial, like their performance status and whether their kidney function is normal, et cetera. But just looking at the very highest level of assuming they were at least screening sufficient to get the NCI match study done, we can now begin to show that it's actually quite much more complicated to represent the information in a clinical trial than ever before. It's no longer a single biomarker with two arms, and we're trying to stratify whether or not that biomarker is predictive of sensitivity to a new targeted therapy. We now have these complicated rules where this is just one of the 21 arms of the NCI match, where now you want to look at BRAF inhibitors and BRAF mutant cancers that are not colorectal cancer, melanoma, or papillary thyroid cancer. So we're applying this new knowledge framework to our manual curation of the clinical trials, and that's going to be in our new version of the clinical trial tool coming out soon. My Cancer Genome content has grown precipitously over the years. I mentioned all of the different assertion types that we now have, but when you begin to think of them from the number of variants that are in all of those assertion groups, the number of combinations of diagnostic rules with the variants that we have is quite an explosive amount of growth, and not something that we expect people to be able to keep up with, which is why we provide the service. To give you a little information on how we go about creating My Cancer Genome, we have a core team shown here. Myself and Christine Lovely are the co-editors in chief, and Christine Michael has joined me here in the room. Christine, just wait real quick. And we represent the core curation team. Now, we have contributors from around the world, over 66 contributors who are scientists or physician scientists or clinicians who are experts in their area that help us hone in on the medical literature that is of greatest value. But our core curation team does a lot of the work of calling that knowledge together to be able to create concise statements that keep with the style guide of a similar language, so that the language is consistent throughout the knowledge resource. But we don't claim that we have all of the knowledge at Vanderbilt, so I always reach out. Please come and speak to us. If you want to become a contributor, we're absolutely thrilled to have you come join us in our efforts. We have been incredibly ecstatic over the dissemination and the wide adoption of My Cancer Genome, starting back from our early days. We've had over 2.2 million page views from our publicly available website from over 200 countries around the world. It's really a national, and actually 50% of those are outside of the United States. And we've had requests to translate My Cancer Genome into 3 different languages. We can't possibly keep up with that request. And we're having about 8,500 site visits a week now, which in the context, there's only about 8,000 oncologists in the entire country. So it's pretty exciting what we're seeing. And of course, we've evolved. We also have a mobile app, so you got to keep up with the times. And so we've had over 3,200 downloads of our mobile app and over 20,000 sessions using that. Everyone's on the go and wants to sit in their tumor boards and look things up as they're going along. But what I'm really, really excited about beyond the publicly available resource is really the clinically integrated solutions that we've managed to do. So when we first started My Cancer Genome at Vanderbilt, we did this out of our own internal need to provide information to our own clinicians on what to do with the data that they were about to receive. This was back in 2010, and it was really revolutionary at the time to be doing this type of multiplex testing. And our clinicians, although world experts, still needed a guiding hand. So we've now done this integration between our electronic health record at Vanderbilt for over 5,500 patients, linking out to My Cancer Genome as sort of an info button resource on a per variant basis. But what I'm really excited about has been this collaboration with GenomOncology, because it's allowed us to take it from being more of an info button type of service to being an interpretive report. Because when you think about the point at which a clinician makes a decision, it usually isn't at the point where they're about to enter in their chemotherapy order entry. That's way too late, right? Your pathology reports that you create really drive a lot of the decision making that the clinician will make at the, well in advance of getting insurance authorization and scheduling the patient and doing all the things that they need to do beforehand. So this is really one of the key points in the decision making process where we can influence what a clinician is going to do and how they're going to interpret the information that they're receiving. And so with GenomOncology's translation of our knowledge base into their framework, we've now been able to use My Cancer Genome content in over 3,200 interpretive reports. This started out, Roswell Park actually addressed us many years ago, was our first group that was interested in using My Cancer Genome with their interpretive report pipeline. And this is a framework that we described where you need knowledge bases at each of these stages of variant identification and clinical interpretation of the variants, and that My Cancer Genome could be used as a knowledge base for the molecular annotation of variants as well as their clinical effect to be able to drive clinical decisions. And so starting back in 2013, we took this approach. And now with the GenomOncology clinical workbench, we've now integrated My Cancer Genome, and this is the GenomOncology order entry screen, their clinical QC validation point, which also uses My Cancer Genome to just sniff over the BAM files to make sure that you haven't missed any variants, and then the clinical workbench, which creates the report that is the final consumption of the oncologist. Just to give you an example, here's a screenshot from the variant calling tool where it classifies the variants according to their actionability in the tumor type, actionability in other tumor type, and not actionable that we know of to help in the variant calling step of the process. And then as you just demonstrated, we have the content is directly coming from these My Cancer Genome assertions that have been translated into the GenomOncology framework to be able to say what the clinical significance of a variant is, both in combination for drug sensitivity, either level 1 in that disease or in some other disease, and also for the clinical trial matching that is available. And I'm pretty excited because these second pages that you show with the additional detail that a clinician can read are coming directly from the content of My Cancer Genome. So it's incredibly easy to create these. You can edit them, but it comes from a source, and there's a complete with a link to My Cancer Genome so that you can, my perspective is that an interpretive report should be made at a time with all of the knowledge that you need at that moment to make your interpretation. But knowledge changes over time. So with the link to My Cancer Genome, if that knowledge changes 6 months later and I'm still using the same assay and the same specimen from 6 months ago to make a decision, although God only knows that's going to be changing radically with rebiopsies and peripheral DNA, now you can link to My Cancer Genome, or the clinician can link to My Cancer Genome to get up-to-date information. One of the things that I'm most excited about is the evolution of the clinical trial representation, and really starting to think about how we can provide some decision support downstream from the lab to the clinician. And we did an analysis with GenomOncology a couple weeks ago of we have over 800 samples of NGS testing at Vanderbilt that was using Foundation's platform. And we ran our cases against the NCI match 21 clinical trial arms to see how many of our patients at Vanderbilt should they have got, what percentage likely would have been eligible for NCI match had they gotten tested today and not a year ago or 18 months ago. And what we were absolutely amazed by was that we had a 36% potential match rate. Now, if any of you have NCI match open at your institution, they've only published the first 10 arms of the trial. Those are the first 10 that are available. And the first 10 have a lot of, how do I say this, FDA-approved drugs that are being evaluated for different indications, like the BRAF example I gave you in vemurafenib. So there's a lot of disease exclusions in this first set. And so in that first set, we only had about a 7% match on the trials. But when they extend it now to the full 21-arm study, we're showing that, generation sequencing for institutions that have this study open likely could potentially have a 31% impact on actionability for therapy for a patient at your institution. Because one of the biggest challenges has been in doing all this testing is you get the results back, but do you have anything at your institution to actually offer the patient, especially for all of these genes and variants that are really more in that translational space and not yet in the FDA-approved companion diagnostic space. So our next step with this is in working with GenomOncology is really to extend our clinical trial annotation and start integrating this into an algorithm that can be used in the clinical workflow downstream of the lab by the clinicians themselves, because they want to be able to search the trials themselves and filter it based on what they think the criteria are most important to them. I'm going to leave you with a few sort of more, visionary kind of ideas. We were talking earlier about targeted therapies and how much promise they've had and how much they've increased the percentage of patients who are responding to the initial therapy when a predicted biomarker is in place. But we still have these patients who are going to have primary resistance to that therapy. Oops. I'm not going to use that again. Who are going to have primary resistance to that therapy, and we don't know exactly what the molecular mechanisms are for why they're having resistance. And then, of course, almost all the patients who advance disease, despite having a targeted therapy and responding initially, will eventually go on to progress. And there's different ways in which the molecular progression is happening for these cases. Well, at any one given institution, we only have a small number of these cases in any given year. And so we really are in a situation where now we need to be able to pool our data together from multiple institutions to begin to understand the outcomes for patients in an aggregate way. And this is a radical thing for us all to agree to share our data, to feel comfortable and secure in that. And it's going to be a massive cultural shift and beginning to evaluate what the value proposition is for each of our institutions to want to share that information. And of course, it's more complicated than that because it's just not about the variants themselves. You need to be able to curate the information on the clinical outcomes and what therapies the patients received and how do they respond. So there's a little bit of good news in all of this is that a lot of that data is now in a digital format where it never had been before because we now have massive penetrance of electronic health records amongst clinicians in the United States. Over 83% of physicians have some basic EMR, but the data inside it is pretty dirty, but it's also very, very deep. So just to give you an idea of the type of data that's created every year in Vanderbilt's electronic health records system, a hospital that's rather large with 55,000 discharges each year and 1.9 million ambulatory visits, 7 million clinical notes get created in that process, which equal about 10 trillion words, which we have to extract from. 9 million scan documents because, unfortunately, many of our patients are seen outside, and we have to get that data back into our system somehow. Over 5 million lab orders, 8 million medications dispensed, 15 million clinical communications, and et cetera, et cetera. And that's just at one hospital. You then add that onto the over 5,000 hospitals that we have in this country, 25,000 clinics, and the 10,000 reference labs, of which many of you are part of. And we begin to have the possibility of finally getting this data together. But we need to be able to be collecting it in a digital format from the beginning, which I'm extremely excited about what GenomOncology is doing across this entire space for the lab world, to be able to capture that data in a digital form, keep it in a digital form in the back end, and even if you create a textual report as the final product, be able to share that information for secondary use. And the real question will become, is there this evolution in clinical decision support coming at us, where we go from our traditional evidence-based protocols to protocol-driven care, and now I don't know how much you're paying attention to all the pressure of pathway-driven care and bundled payments and all these things, and will we ever be able to get beyond that to a data-driven form of clinical decision support? And it's these infrastructures that we're creating, starting with getting the data in the right format from the beginning, and then bringing that together with other forms of information that are collected as part of the routine treatment of patients downstream, and bringing that together to do data-driven decision support is really going to be an amazing opportunity. So with that hopeful future, long way to go to get there, albeit. I just want to acknowledge the My Cancer Genome team and all of our collaborators at Vanderbilt and our contributors worldwide, and the GenomOncology team who we've been working together with so closely over the last 2 years to really bring this to fruition and bring the next generation of My Cancer Genome to a disseminated mass we never could have imagined. So thank you for your time, and I think we were both going to take questions together.
It was free, isn't that wonderful?
I think it's my credit card. Oh.
Oh, such a mass exodus. Can we get these turned on?
Regarding the multimodalities, Dr. Peiper, like immunohistochemistry, NGS, would you perform them simultaneously or sequentially, or is there an algorithm? Or
So for most immunohistochemistry, we've done, if we couldn't do sequencing, then we do immunohistochemistry. We consider sequencing better. The ALK story, I think, will be interesting, and I don't want to lift the kimono too much, but about 40% of patients who have ALK rearrangements by FISH don't respond to crizotinib. And we have a few cases that have KRAS mutations, and the immunohistochemistry is interesting.
I think, and I'll just speak to that point as well, is that there have been a lot of different frameworks coming out with, should you do sequential testing, should you do an entire panel but sequentially report, and when they're sequentially negative, reflex to now I'm going to report the other ones and sort of charge on a rolling basis. So that's been one of the models that GenomOncology has been supporting also. And then at Vanderbilt, we've done a diagnostic management team approach in the hematologic malignancies, where we kind of let the pathologist drive a lot of the sequential testing based on what they find initially and that evolution. So it's been very interesting.
Ethical concern that you have generated the information but you're not releasing it?
There's always that concern, but there's also the concern of giving too much information and billing for something that doesn't have clinical utility in that setting. And so it's a complicated story right now, and what we can bill for and what we should report is still absolutely, you're asking the right question.
So, yeah, that's a complicated issue. So we just last week had a case of a melanoma, actually a palatal melanoma, where they ordered a BRAF mutation and then studies for resistance. We'd done since it was for resistance, we'd done panel sequencing. The BRAF had been negative, and it turned out, as you would expect with a mucosal melanoma, it had a KIT mutation. And so we asked the clinician to order that test. They ordered it, and then we sent out the report.
Thank you for a great talk. So just from a logistic point of view, for the small biopsies to the immunohistochemistry versus sequencing, so you go ahead and cut unstained sections before you attempt?
So our reflex is that we cut unstained sections that are when the block is faced up, because otherwise, as you know, with a small specimen, if you have to reface, you lose. If it's small, we're kind of on alert, but we look at the yield of DNA, and if the yield of DNA looks low, we just use the unstains for immunohistochemistry. Yeah.
Thank you very much. I'm Mike Dugan from Exact Sciences. How much consideration do you give to whether it's primary versus secondary malignancy? And kind of go back to your textbook example, Dr. Peiper.
Yeah.
When in the old days, the clinician didn't really care whether it was non-small cell squamous or adeno. They just said, tell me whether it's small cell or non-small cell, because I only have 2 drugs. Now they have conventional chemotherapy and the targeted agents, how much consideration do you give, whether it's primary or secondary, before you get into all this nomenclature about the mutations and so forth? For the
So we
targeted
typically
therapy?
do TTF1 and napsin-A. As you know, it's not perfect, and so we've had cases where they've ordered the test, the clinicians have ordered the test even though it was a squamous. But I think we're off the hook now with the new ASCO guidelines. Because the new ASCO guidelines say if you can't exclude adeno, then go ahead and do the testing.
But what if it's metastatic?
Well, I mean, there we have to go by the clinical diagnosis, and we tend to err on, if the clinician wants the test, we tend to err on the side of doing the study.
I only say that I used to work for Biotheranostics, and there were a lot of, there was a very good study showing that if you got the right primary, you could improve survival. And if you didn't get the right diagnosis,
Sure.
of course, you're left with just palliative therapy. But for some tumor types, there's like breast, for instance, there's a really dramatic increase in survival if it's treated with a breast protocol versus a
Right.
lung protocol. So then the question becomes, have you really spent the tissue wisely to go directly to all the next generation sequencing when you might have missed something more obvious right up front?
Well, it depends. Is it a small biopsy? Is it not a small biopsy? If it's not a small biopsy, you haven't done any harm. If you're consuming tissue and you're not certain, and there's no other known primary, it's one of those cases where you do the best you can do, right?
Yeah.
I think in a way the ASCO will facilitate, because we spend a lot of tissue trying to decide adeno or not. So if this has become more relaxed, well, those additional sections will be used wisely. So I think that's a good move, and also for earlier stage disease.
Okay. Well,